I'm just doing a sound check. Does this work? Okay, very good.
right there.
Okay.
Okay, we should be good now.
So sorry, our
you're
online
fine.
friends. Um,
Should still be in present. There we
okay.
go.
So sorry, our online friends, you missed a little poem.
So anyway, about me, formal education in packaging science, and I work full-time in manufacturing actually, but I spend all my free time in Louisville, North Carolina on a hemp farm. If anyone's ever heard of honeybee hemp farms before, I started working with Alan Pickett, that owner, a few years ago. I joined an online program called the Soil Food Web School in 2020, and that's how I've learned and trained and all the expertise and all the things that we're going to talk about today. I'm currently in their Soil Food Web Consultant training program. So hopefully in the next few years, I'll be out in the field improving yield using biological amendments with agricultural producers, private gardeners, landscapers, things of that sort. And then I started my own company last year called Salem Soil Solutions. And right now I do produce what's called BioComplete or BioComplete compost, which is a compost that has the full web of living soil organisms, which you'll see a lot about today, as well as worm castings, which is just worm poop. Um, so there's me without a mustache on the hemp farm. So this is the woman that started the program, Dr. Elaine Ingham, and that's her and I at a composting workshop in Costa Rica last year. And so Dr. Ingham has been an integral part in the world of soil biology for over 40 years. She is what some people would call the grandmother of biological soil sciences, which directly oppose a chemical view of soil science that just breaks soil down into molecules, chemical compounds, and its constituent textures and structures, which is how most conventional agriculture is done today. Her methodology is being used all around the world. It's spurred hundreds, if not thousands, of companies that are devoted to restoring soil biology, which, as we'll talk about today, has a direct impact on the global and local ecosystem of the world. So an outline of today's talk, we're going to define soil, what's in soil and what makes soil soil. It's the biology. It's not just the organic matter. It's not just the salts, the clays, the silts, things like that. And then we're going to talk about the benefits, why you should think about the soil food web when you interact with your plants or garden, and also some ways to strengthen the soil food web or to inoculate your environment with these soil food web microorganisms, specifically through compost, compost extract, and compost teas, which are a liquid form of compost, and vermicompost, which is composting with worms. Then we'll do a little question and answer at the end. So what is soil? There's one difference between or there's a couple differences between these two definitions, and scientists will still define soil differently. So as defined by the father of soil science, we're going to need the parent material, which is the sand, the silt, and the clay. And all these materials are going to be properly balanced. I'm going to show you a soil texture triangle next, which a lot of you have probably seen before. You're also going to need organic matter. Does anyone know what organic matter is? So there's a lot of it piling up on the curbside right now. It's leaves. So anything organic that falls in the ground, whether it's a dead animal, whether it's like in the poem, it's insects, organic debris, falling trees, leaves of that sort. And then as defined by the Green Revolution, the conventional farming philosophy that took over in the 1960s and has led to a massive interest now in soil health, and in my opinion, has led to massive global warming. We're going to only look at soil from its mineral component. It's going to be completely organic. We're going to look at nitrogen, phosphorus, potassium, and then we're going to look at rock, essentially. So here is the father of soil science's view of soil. Over there on the left, we have a soil PED, which is just a cross-section of a soil horizon. And at the very top, this is where all the action happens. Does anyone know what that O stands for?
Nitrogen.
Organic.
Oh,
That's the
organic.
organic matter horizon of soil. It's going to be anywhere between 2 inches and even up to a foot deep in some instances. A lot of regenerative farms around the world are actually building several feet deep of organic matter to plant straight into. But here in the Carolinas, most of you are familiar with the A and the B horizons, where we get that Carolina clay. When they're doing landscaping on campus, you all see it. It blows up like it's the Dust Bowl. It gets stuck to your hands, things like that. And then at the very bottom of the soil bed, we just have the bedrock or continual parent material. So the O horizon, depending on where you are, can be made of any form of organic matter. Is it standing dead from harvested crops? Is it cover crop planting? Is it clover? Is it right now leaves? So again, as defined by the Green Revolution, it completely voids the last slide that I just showed you. It's only going to talk about sand, the silt, the clay, and then its constituent mineral parts. So in conventional farming, the soluble minerals must be added to the quote unquote inert support matrix, which is their definition of soil, in order for plants to grow. So here's more of a classic view of soil. Maybe the home gardeners have heard of a loam before. You want your soil to be loamy, but that's made up of 3 different sizes of mineral particles: sand, silt, and clay. Sand's going to be the largest, silt second, and then clay is going to be the smallest. And then here's that soil texture triangle for you, where if you have all these minerals perfectly balanced, you end up with a loam. If there's a little bit more clay, you have a clay loam. If there's a little bit more silt, you have a silt loam. And these are really just broken down by size of particle. So one thing that's fascinating to me about clay is how small it is, just because you see so much of it. But under a microscope, each clay particle is about the size of bacteria, and they're hard to identify. But when you get tons and tons and tons of those particles together, you end up with those big, huge red dunes outside of a landscaping or building construction situation. So this is a natural view of soil structure. So soils all around the world have different structures. They look different. They feel different. They're largely curated by greater environmental factors. They're curated by how the planet has evolved over time. And there's a bunch of different types and names, but we won't go into those today. And then we have more of so this would be Mother Nature's baseline view of soil structure, but this is the human view of soil structure, or how we're artificially changing that soil structure. So over here on the right, we have soil with conventional tillage. I don't know if you can recall that soil horizon that I showed you, but it looks like that C part at the bottom of that soil horizon, if you recall, gets really close to the soil surface. And zooming in on that photo, those plants don't have much room to grow. We barely have an organic matter horizon. And in some agricultural settings, we don't have an organic matter horizon at all. Let's say we go into a reduced tillage situation. We're going to get more organic matter. And then a no-till, it's been a very popular buzzword these days in regenerative ag. We're going to have a huge organic matter horizon. Over here on the left, you're going to see some blue stem. And this is what plants are supposed to look like under the soil surface. Look at how deep those roots go. Isn't that unbelievable? I remember several years ago now, it was the first time I was just stunned and floored at photos like that. Grasses on the plains, in the Midwest should really be dozens of feet deep. There's some species of grass, they really don't know how deep they can go. And so again, just to go over these, we're going to look at soil structure, which has a direct relationship with root infiltration. So in a situation where we're not tilling at all, we're not disturbing the soil, we're not breaking up any of those micro and macro aggregates in the soil, the roots are going to go plenty deep, right? And then we're going to bring that plow by and we're going to break up some of that soil structure. And after we plow enough times, we're actually going to end up with a compaction layer right here. So if you till your field enough times with that mechanical instrument, at the bottom of that mechanism, you're going to start forming a concrete barrier. And I use the
Wow.
word concrete because it's relatable, but you can actually compact clays. In North Carolina's heavy clay. You can compact clay to a higher density and a lower porosity than concrete. So if you go out into, I don't know, 30, 45 minutes west of here into Louisville, there's compaction layers from tobacco tillage under the soil just a few feet deep that are actually denser than the sidewalk that you walk on to class, which is, to me, unbelievable. So zooming in, we're going to break out the microscope. I'm sure most of the students have seen a microscope before, but if you haven't, this is a compound microscope. So there's a light that comes from the bottom. It goes up through a lens here. And then I don't have my objective lenses attached because they're quite sensitive. I was afraid we might have a mistake today. But then you're going to look through the eyepieces, through the objective lens, and onto your slide. And so this would be a picture, and the subsequent pictures to follow are going to be pictures of soil under a microscope. So those aggregates, that soil structure that I just described to you, this is a 400 times magnified view of soil. So as you can see, there's empty spaces here where light's coming through. There's organic matter debris probably, but all of this is sand, silt, and clay, and it's held together somehow. I wonder what reason all of these molecules decided to stick together. And so here's an electron microscope view of what you just saw beforehand. This is not my photo, but it's just another way of looking at soil. And so we have these aggregates that are cemented together somehow, and it's by biology. We'll get into that in a second. And then we have voids. So, soil structure is extremely important because these voids are filled with air and water, two things that your plant roots need. Here's another electron microscope view of what's holding and what's causing those soil aggregates to stick together. These would all be, I think these are like some kind of bacilli, so rod-shaped bacteria. Bacterias are sticking them. You've all heard of the 5-second rule, right? The reason why there's that commonality there is because the 5-second rule is pretty much saying if you pick up the object quickly enough off the ground, then the bacteria won't have enough time to form glues and stick to that object. But you want bacteria forming glues and piecing those sand, silts, and clay together. Otherwise, our environment would just be loose dust, and we end up with a dust bowl. So this is a colored electron microscope view of soil, and there isn't a space in here that isn't completely covered in biology. So here's a nematode. Bacteria. They're absolutely everywhere. So going back to those definitions, what is soil? Is it sand, silt, clay? Or is it sand, silt, clay in a relationship with biology? I think it's the latter. So the soil food web is what keeps all those aggregates together. They're making soil structure. They're turning dust into a sponge. If you go over here, we have our plants. Everyone knows photosynthesis. But these plants have formed a relationship with this host of microorganisms. And what the plants are doing is they're sending these root exudates, they're sending their sugars from photosynthesis out into the rhizosphere, out into the root zone. And they're telling these microorganisms, hey, I'll trade you some sugar if you could just mine me 6 parts per million of boron. I'll trade some sugar with you. And so going up the food chain here of the soil food web, we have organic matter that these fungi and bacteria are mining for their nutrients to take into themselves and also for the nutrients to trade with their plants. These fungi and bacteria are being eaten by protozoa, nematodes of a bunch of different varieties, and then subsequently larger tertiary predators. But fungi are eaten by, things like microarthropods, which are microscopic bugs. They're eaten by fungal feeding nematodes. They're eaten by bacteria in certain conditions as well. Bacteria are eaten by protozoa, which are single and multicellular organisms that just run around the soil and graze like cows do on bacteria. The bacteria are also eaten by nematodes, bacterial feeding nematodes, omnivorous nematodes. There's nematodes that just have a huge tube at the end of their mouth, and whatever gets in there, they eat it. So spores, pollen, things like that. And then those animals are subsequently eaten by larger predators as well, like microarthropods. And then we all know those bugs are eaten by, birds and mammals. So the importance of this chain is that when a protozoa eats a bacteria or eats hundreds of thousands of bacteria a day, they're going to need to use the bathroom right next to the plant in the plant zone. So what happens is we have plant available nutrients that happen around the root zone that's driven by what's called the poop loop, or a predator-prey relationship between these higher level predators and these lower level microorganisms. So there's me with a microscope. That's where a lot of these photos that are coming up next come from. Bacteria. So bacteria, all shapes and sizes. Some are good, some are bad. We've all heard of probiotics. Most of the probiotics on the market actually come from soil microorganisms. All different shapes and sizes. Most of them are beneficial. So cocci, bacilli, and then some of them are disease causing. So like we have some Vibrio here, some spirochetes. We see a lot of these pathogenic organisms of bacteria if I run a compost tea brewer too long and it starts to suffocate. So our good microorganisms always breathe oxygen and they're always safe for human contact and even consumption. Our bad microorganisms are organisms that develop out of a low oxygen or a no oxygen environment. They'd be anaerobic organisms. So this is a picture of bacteria. Really interesting. All these little dots everywhere are bacteria. If you remember that electron microscope picture that I showed you, that they're covering everything, this is probably like a 1 to 1,000 dilution of soil. So 1 milliliter of soil and 1,000 milliliters of water, and there's still bacteria everywhere. Here's a much closer view. This is probably 1,000 times magnification. These aren't my photos, but here's a streptococci. So, next, everyone's favorite in the past few years, fungi. I'm surprised at the amount of common knowledge that the average person has about fungi all of a sudden, just because of medicinal properties involved with the human body, and then also changes in palate and visual aesthetic. They're quite popular in art these days. They're quite popular as supplements and on your plate. But what's really important for soil biology is what's happening below ground. So some of you may know these are all the fruiting body of different fungi. So these are the reproductive structures of the mycelial network, which lies below ground, that you can see with the naked eye if it's established enough, but you do need a microscope to really quantify how much of it is there. I took all these pictures. So just a little bit of information on the fungal reproductive cycle. We have a fruit body, it drops spores. Those spores germinate, it turns into the mycelial network. The mycelial network turns into a fruit body, it drops spores over and over and over and over again. So this video should play. I want you to look. I don't know if we have enough good lighting in here, but these jack-o'-lantern mushrooms are actually all dropping spores. And I was walking by when the light was hitting it just right. And it's one of the first times that I had seen it at the time. And it's just amazing how much. So a lot of these fungi have evolved to drop this many spores because the reproductive success is pretty low. So 2 of those spores just have to find each other magically in the environment, and they germinate, they make a new mycelial network. So where do the rest go, right? They feed the soil food web. All those microorganisms are dining on the leftover spores of these fungi. So Here's just a cross section of the mycelial network running into pins or mushroom primordia. And back, we're thinking about soil structure. Think about this mycelium's creating a big net for all those sands, silts, and clays and holding everything together. I'm not an expert in endo and ecto mycorrhizal fungi, but they are super hot right now in the soil food web discussion as far as plant growth, yield, and resilience. So these are plants that have been colonized with fungi that have a direct relationship with the cells within a root tip. And if you could think about the average root cell or root hairs, and then look at all this surface area that the fungi is giving these plants. And so anything that endo or ectomycorrhizal fungi is going out and mining, as far as nutrients go, it's giving it to the plant. Some people also believe that that fungal network is allowing plants to communicate with each other in a lot of different ways. So here's a nice textbook view of that arbuscular endomycorrhizae and ectomycorrhizae. So endo meaning inside, ecto meaning outside. So there's types of fungi that get into the plant cell root tip. So here's the root tip, right? And they're directly colonizing root cells within the root itself or outside of the root. And then think about all that extra surface area that plant now has to go get all the nutrients that it needs. These have completely disappeared in a conventional agriculture setting. They no longer exist. And it's because of the pesticides, the fertilizers, the fungicides, the chemicals, things like that. These mycorrhizae do not have fruiting bodies a lot of the time. So you're not going to be able to see them unless you pull a root out of the soil, you stain it with a staining liquid, and you examine it under a microscope. So here's another view from the top down of that relationship. So we have fungi going into the root tip, into the cell wall, and growing out, and they form a symbiotic relationship. Trichoderma. So you all get this on your bread, and it's just another way of saying that you're covered in biology all the time. If you do nothing other than put bread in your cabinet, suddenly you're going to have this green bloom. Everything's covered in spores all the time. It's just a matter of the environment to be just right for things to wake up. Right now in conventional agriculture, there's a lot of tests showing promising results with just adding trichoderma to the soil. So just adding one species and group of a fungi to increase plant health and yield overall. So this is fungi under the microscope. This is a picture I took. And so all these branches, this would be considered like a mycelial waft, but then you can also look at the individual hyphae, so the individual cell of mycelium right here. So these are septa, these little cross sections here, and that represents one fungal cell. And I think this is a video. When you look at a microscope, you're looking at the height of the drop of water on the microscope. So as the focus changes, we're actually we're going up and down the view of that fungal waft there. This magnification would be 400 times. So total width here would be several hundred micrometers wide for this field of view on this screen. Going up the soil food web chain, we have amoebas, flagellates, and ciliates. So these are our bacterial grazers. They're keeping the bacterial population in check, and they look quite pretty as well. I couldn't get a great picture of a flagellate, so I had to pull some online because they're just too fast when they're alive. So the reason why they're called flagellates is because they have this tail that's called a flagella, and they use it to move, groove, and bumble around. These would be amoebas. So there's a bunch of different species of amoeba, especially soil amoebas. We have Arcella, We have testate amoebas. This is generally a common image when you think of an amoeba. This is a naked amoeba. So this is an amoeba without a shell, and it has those pseudopods, sticking out, those false feet. Here's a amoeba under the microscope with soil, next to a fungal strand. These are photos I took. So here's testate amoeba. And again, right now, the amoeba is actually really upset because I put it in a microscopy sample and I shook it up in some water. So the amoeba's gone back into its shell called a test. And the tests actually have a really great structure to them. It's made up of these really, really tiny hexagons. So we see that shape all over nature, honeycomb, things like that. So here's an amoeba, here's an amoeba down there. Some additional photos of amoeba. Soil aggregates being held together by fungi, bacteria. And then these are ciliates. So ciliates are called ciliates because of their cilia, which are these little tiny hairs that they use for locomotion. So they're really quick. I couldn't get a video of these either clear enough to come across the screen just because they're that fast. But we see high proportions of cilia in compost tea samples that are trending towards suffocation or trending towards anaerobic. So if, let's say, you're a gardener and you decide to make compost tea and you want to know whether your compost tea is good or not, you have a new recipe, you've been brewing it for 24 hours, and I take one look at it under the microscope and I see a ton of ciliates, I'll say, okay, you brewed it too long or something's wrong because your tea's suffocating. Nematodes. So here's an electron microscope picture of a nematode. This statistic blows my mind every time. 4 out of every 5 animals on the earth statistically are nematodes. Crazy. What's that in the top left? Yep, 57 billion nematodes for every human on the planet. So they are everywhere. Nematodes have a bunch of different feeding groups that can be identified using, basic microscopy. Bacterial feeders have a vacuum for a mouth. Fungal feeders have a spear in their mouth, so they can puncture that cell wall that I pointed out to you earlier. Plant parasites, when gardeners or conventional farmers hear of nematodes, they're only talking about this group. This group's giving them all a bad name. They have a spear and basal knobs. And so these knobs are the muscles that allow that spear to go out and puncture through a cell wall. And they're what's giving your plants root galls and plant disease, because your plants, like I said, there's a lot of nematodes. So in a conventional agricultural situation that's compacted, it's suffocated, there's going to be tons of root feeding nematodes attacking your plant roots from all angles, all day long, all year long. There's omnivorous nematodes. So they eat a little bit of everything, whether they spear a piece of fungi or they decide to eat some bacteria, they decide to eat some spores. There's also predatory nematodes. These are just so hilarious to me. The predatory nematode has a giant tooth in its mouth. Can't miss it. And what's unique about the relationships between these nematodes is that the predatory nematodes actually prefer root-feeding nematodes because they're really slow. So they'll seek out root-feeding nematodes first. So if you have a root-feeding nematode problem, your solution isn't a nematicide that's going to take out all your good guys and probably affect other biology. Your solution is to actually reintroduce predators into the system. And we've seen that successful at a much larger scale. If you think of the wolves re-entering Yellowstone, suddenly the riverbanks aren't eroding anymore because it's controlling the elk population. We don't want to control your plant disease with synthetic chemicals. We want to control your plant disease with a biological solution that it lacks. So this is a video, and this is what would be called bioturbation, or movement of soil through biological means. If you think of a worm going through the soil, that's bioturbation. But as I've discussed, there's tons and tons and tons and tons of nematodes. And so if you think of this movement action across all living soils across the planet, you're going to get a tremendous amount of soil movement in every single micrometer of soil on the planet. Compare it to like hitting one of those door stoppers. They're like springs. This one's a bit slower. This is a bacterial feeding nematode. I might have stunned it with the lighter. And you can tell it's a bacterial feeding nematode because of some of its organs on the inside, but then also it has fancy lips, is what we microscopists call it. And so I could not distinguish the different species of bacterial nematodes. Of that, there are thousands, but I could tell you if it eats bacteria, and a bacterial feeding nematode is going to be a good nematode. Here's another bacterial nematode. So it's just got that vacuum for a mouth, and then it has a terminal bulb. So this is the muscle that it's going to be used to pull in bacteria. And if you look inside of the nematode, this is all bacteria. So just like worms in your garden are taking in an organic matter and then pooping out worm castings, which are nutrient available biological fertilizers for your plants, think about all those nematodes in your soil that you may or may not have anymore, depending on your farming practices, are all fertilizing passively day and night, year round in the root zone. Additionally, they're keeping those bacterial populations in check. And due to the research that Dr. Ingham has done, her and her husband, they found that bacteria behave completely different in an environment that has predator pressure from nematodes. Another bacterial feeding nematode. There's a testate amoeba, soil aggregates, bacterial feeding nematode. This might be the last one, I promise. Okay, there we go. All right. Working up the food web, getting into larger critters that you can see with the naked eye. So here's a bit of a scale for you on size. We're going to move into microarthropods, little tiny spiders, white flies. Everyone recognizes, something like this at some time in their life, whether they know what it is or not. I couldn't tell you the species either. But then here's a microarthropod under a microscope. So these are going around just like cows and grazing those nematodes, grazing soil particulate and digesting the bacteria within them, grazing fungal wasps and digesting the material within them. And then they're leaving behind plant available nutrients in and on the root zone. They're also keeping pathogen organisms in check. I love this picture just because what is above is so below, right? So we think, I talked to one of my mentors about this, and she thought that there's a microarthropod with a feeding mandible of some kind, and this is a 400 times magnification, but look at that lobster-looking claw from a microorganism. So they're not only grazing around the root zone, but they're also your biological taxis for the part of the plant that sticks out of the ground. So when those microorganisms are grazing on the ground, they're also getting picked up by other insects. They're making the way up the stem themselves. They're flying from the ground to the plant itself. And then we start to get a biological activity in a biome on top of the leaf and plant surface. And that's going to have amazing effects for the plant, depending on what kind of biology you get on the plant. So we would call those microbe taxi cabs, the microarthropods. This is actually a diagram of dung beetles. You're in North Carolina. There's a lot of grazing here in North Carolina. What they found in regenerative grazing operations is that the indigenous populations of dung beetles are coming back, and they do not exist if you're giving your livestock antiparasitics. So that those chemicals and pharmaceuticals that you're feeding your livestock are making it through the ruminant animal, making it through the digestive tract, and they are demolishing the soil microbiome and organisms that their manure lands on. So it's very, very important to have things like dung beetles in a system with livestock because they're going to go up, they're going to mine that organic matter, they're going to come down, they're going to use it to reproduce. And so not only are they doing a mechanical degradation of this manure, but they're also doing a chemical degradation of this manure and leaving behind plant available nutrients that they've digested and put down themselves. But think about when it rains now. Look at all of this surface area for water to collect. So suddenly your soil structure's changed, your water retention's changed, your plant available nutrients have changed, and this manure patty is going to go away. So it's going to get digested. It's not just going to be sitting there in the field like it is in a lot of conventional livestock operations that use chemicals and don't have any biology to do the labor of breaking down that organic matter. I've thought about this kind of as like a rain cloud falling. So we're going to talk about worms a little bit later, but there's an important difference between your average American and indigenous earthworm population that you see maybe when it's raining on campus and they're on the sidewalk, or you're on your farm and you're walking down the road and there's night crawlers sticking halfway out on the ground. So these earthworms burrow, and they burrow quite deep. They don't want to be around a lot of other worms. They kind of have a turf. And the different species burrow different directions, some more horizontal, some more vertical. And earthworms really like to eat post-digested organic matter that's going to get digested by microorganisms. So they'd really prefer to eat soil. And that's going to be a lot different than the composting worms we see later on. But these worms are also affected by whatever chemicals you're putting, whether in your livestock or on your plants, and they will disappear or die out if you put the wrong thing down, especially at the wrong time. Invasive species warning. This is a hammerhead worm. I believe these are from Asia. Their slime is toxic and a skin irritant to skin, eyes. If you see one of these, do not eat one. Not that you would, but I've seen a handful of these in the farm lately, especially in my compost piles that I put worms in. They prey on earthworms. They kind of turn them into ghosts and suck out the insides. And so if you see one, do not smash it, but you can pick it up with a bare hand as long as you wash your hands later. But go ahead and pick these up and just put them in a Ziploc bag and sit them in the sun to dry out. If you try to smash them, they will regenerate from their broken bits. And then you have it's like a hydra. You'll have 5 or 6 of them reproduce. So if anyone sees these, be a good citizen scientist and go ahead and take them out of the environment because they are harming indigenous earthworm populations at this time. So benefits of the soil food web: disease suppression, nutrient retention, bioavailable nutrients, toxin decomposition, and they build and hold soil structure. Disease suppression. These microorganisms are building a castle wall both on your plant surface and in the root zone. So a pathogen that wants to come feed on your plant cannot get to your plant. If you remember that electron microscope picture that was really colorful, if air-breathing beneficial microorganisms are in the way, your plant cannot get preyed on by pathogens if there are beneficial organisms in the way. And unfortunately, most conventional agriculture settings and even chemical-dependent home gardener settings are void of most beneficial biology. So they've succumbed to really weak pathogens in general. Forget what the next one is. Okay, retain nutrients, runoff, and leaching. There's a lot. This is a little science experiment, that an elementary schooler might run. On the left, we have soil with plants. In the middle, we have some organic matter debris, and on the right, we just have bare soil, and then they run water through it. So if you look on the far left, the soil with living roots, the water is going to come out clean. With just a little bit of organic matter debris, the water isn't as dirty as just bare soil. So we have massive amounts of runoff, which takes away all the nutrients from the soil. So if you fertilize with a basic NPK, a salt or something like that, when it rains, most of it's getting washed away. Most of it's leaching out. And not only that, the biology, by virtue of the fact that it exists, it has to be made out of something. So those nutrients that are in the soil that feed and allow those biological organisms to live and exist, because that organism is alive, when it rains, it doesn't get washed away, and the nutrients that it's made out of do not get washed away either because they're locked up inside of a living organism. So there is no soil on the planet that cannot support life. These are chemical element values for mineral soils across the planet. There is plenty of essential micro and macronutrients for all plants in sand by itself. But the only thing that's missing is the biology, the fungi, the bacteria to go in and mine these essential plant nutrients out of those parent materials and then get eaten through the action of the soil food web and then disposed of through the poop loop in plant available form around the root zone of plants. Recently, this has become pretty popular in like health food circles. This is the change in nutrients. Which one is this? In vegetables since 1940. A lot of naturopaths right now are saying, okay, we have all these autoimmune diseases, we have all these cancers, we have all of these, we have this neurosis in late age because our bodies are completely void of essential vitamins, minerals, and nutrients that are no longer in our vegetables. The reason why they're not in the plant is because they're not being delivered to the plant in nutrient available form. They can't get nutrient available forms of these minerals unless there's that predator-prey relationship in the root zone. So this would be vegetables since 1940, and this is fruits since 1940. So I don't know if anyone's tried, I don't think magnesium's on here. I know personally for me, taking a magnesium supplement like changed my life in 2 weeks. So this is, excuse me, retains nutrients, bioavailable nutrients, enhancing nutrition for plants and animals, and then toxin decomposition. If you've had Roundup use in your garden for years, it's okay. Once you reintroduce, especially fungi to that system, they're going to clean it up. These are experiments run by Paul Stamets, a renowned mycologist, and my original inspiration foray into mycology at the time, several years ago. He did an experiment on a crude oil laden kind of piece of parent material. And within just months after inoculation with a simple oyster mushroom, oyster mushrooms created a flush within that organic matter pile covered in crude oil. The fungi cleaned up and synthesized that crude oil into bioavailable nutrients. The birds decided to come and eat those oyster mushrooms that were fruiting out of that pile, and then they dropped seeds. And because all the crude oil was gone, because the fungi cleaned it up, those seeds decided to germinate and grow. So here's another example of that crude oil into a mushroom substrate. The fungi really know what they're doing. They're amazing chemists, and they will, the term is called bioremediate. The fungi will bioremediate almost all, well, some people would say all organic compounds in the world. So whatever chemicals you're putting down, given enough time and given enough biomass of fungi, especially a diverse array, they'll be able to clean anything up. And then we have soil structure. So this is a picture from the Dust Bowl. This is when conventional tillage went overboard. We got rid of all soil structure, and we decided that we wanted aesthetically pleasing, completely flat, not a single living root fallow field way back in the day. And that led to a die-off of all biology. And when there's no biology, there's no glues, there's no glues from bacteria, there's no fungal nets holding those micro and macro aggregates together. And then when the wind comes by, it blows all the topsoil away. This is what should be happening in a living soil system. So when I say the term soil sponge, it's not only the soil itself being a sponge with plenty of room for oxygen and water, but when you add living roots to it, think about a base flow of water running through roots like that. Surely they'd come out the other side clean. And then here's conventional agriculture. There's probably a compaction layer here due to tilling, and those roots aren't going very deep. So in a conventional agriculture setting, and then in your gardens, if your soil's compacted and your roots can only go this deep, what happens if it doesn't rain for 2 weeks, right? There's plenty of water down here for healthy soil. There's plenty of water down here from great water infiltration due to healthy soil. And then there's plenty of root depth to come down and get water during a drought. But in this situation, you're going to have boom bust crops. One day your plant's going to be really happy because you just watered it, and then a few days later, it's all wilted. What just happened? Did it get too hot today? And the reason isn't your watering practices per se. The reason is not only do your plants not have a three-dimensional substrata to access for their nutrients and water, their water uptake, but they're actually going to start competing with each other because the compaction is going to be so high in this plow pan that those roots are going to go down and out. So how do we restore the soil food web? One way to do it is great compost. I would love it if some students took on a big composting project. Let me know if you do. I'd be happy to come help out. So there's a bunch of different ways to compost. It depends on your feedstock. It depends on the level of effort you want to put in. It depends on your environment. It all depends. It's very gray. So in a few short slides, I'm going to talk about specifically thermophilic composting, which is composting with heat. And so that's going to it's going to determine our feedstock materials. It's going to determine how we maintain moisture and monitor temperature. And then we're also going to have a protocol to follow for cool down and maturation of that compost pile. So this is probably 200 gallons of soil, and it is a mixture. Oh, equipment. All right. So if you want to make a thermophilic compost pile at home, you can do it on the ground. You don't have to do it in a cage, but there's a really great reason why we do it in a cage, and it's for airflow and water evaporation capacity. So we want aerobic organisms that breathe oxygen. We want this entire surface area of our compost pile to have oxygen exchange capabilities. We want water that is trapped to escape because this thing's going to heat up. You could get a compost pile hot enough to explode or catch on fire if you really wanted to. So you'll need a cage, some chicken wire. I've seen a lot of people have been using this product called a GeoBin recently, which is just a plastic sheet with holes cut in it. It's the same as chicken wire, but probably more expensive. You're going to need a pitchfork, a composting thermometer. A composting thermometer isn't like your average cooking thermometer. They're anywhere from 4 feet high to a foot long, and it allows you to get to the center of that compost pile and assess the center of the compost pile's temperature, which is where all the action's happening. You also need water, which is really important and will make or break whatever kind of compost or vermicompost you're making. You also need to cover that pile up because we don't want perfect moisture content in your compost pile, and then we have a rainstorm, a thunderstorm, and suddenly it's at 90% moisture capacity because you didn't cover it with a tarp. Composting ingredients. Really common way to talk about composting ingredients that a lot of you might have heard of is browns, greens, and high nitrogen material, which is our microbe party food. To really get into the weeds, pun intended, we have high nitrogen green material. So materials that are green like alfalfa and clover, but have a lot of nitrogen packed in them, and they tend to act somewhere in between like greens and high nitrogen. So what these ingredients are doing is you're getting them from the environment. For the pictures I've showed you before, they're covered in biology already. and you're going to put them together in a large mass. And what they're going to start doing is going, wow, we have accessible food, accessible water, and accessible oxygen. It's time to reproduce and move around. That's what's going to generate the heat of your compost pile. And what really starts them off is this high nitrogen material like manures. I make compost out of beer mash from Incendiary. It's free. Blood meal, feather meal, cracked corn, so seeds, things like that. If you're a home grower or you have a friend that has livestock or something like that, just be careful with manures these days. So if you have a friend with horses and they spray their hay, or they buy hay that's sprayed in broadleaf herbicide, that horse is going to eat that hay. It's going to concentrate that broadleaf herbicide, and then you're going to decide to use manure to make compost out of with active broadleaf herbicide, and it doesn't get broken down by the composting process because of the way that they've really intensified these chemicals over the years. And then suddenly all your tomatoes die when you put your compost down. And it's because that horse ate hay with broadleaf herbicide. Same goes for cows, ivermectin, and horses for that matter. So they're going to kill all your nematodes. If you have a compost with no nematodes, it's because, and you're using manure, it's because of the pharmaceuticals in the livestock. You don't have to worry about that with organic inputs like feather meal and cracked corn. So greens can be anything from lawn clippings to food scraps, coffee grounds. Even though they are brown, they act like a green material. If you can think of coffee grounds being a coffee bean, like a green bean, but coffee's just brown. And then browns would be any kind of organic matter. So wood chip, mulch, brown leaves, straw. I've heard hemp hurd recently is a great substrate to grow fungi on in general. And so I'm going to try it in some compost piles coming up, but I will have to buy it. Optimally, you would get all these things for free or from your friend. Maybe trade them some finished compost after. So here's a visual on that really basic compost recipe. Depending on the strength of your manure, how high nitrogen is that high nitrogen? All these ratios are going to float, but that's up to you. That you're a citizen scientist. You're going to have to do some experimentation in your backyard. You're going to have to monitor with your compost thermometer and decide what ingredient ratios are right for you. But this is a great starting point. So where can I find free feedstocks right now? Great stuff, the leaves. So this is what I made my last compost pile out of. So these are crown beard. This is just growing in some fields on the farm that I work with right now. And then we blended those down with a weed eater. You can see that there's a little bit of gray and brown organic material in here. So it's not, when we go through with the bush hog and cut all this down, it's not 100% green. So I'm going to factor this in to my recipe and say, okay, I need to add 40% of my green volume because there's a little bit of brown material in here. Wood chips, this is really going to be your structure and your housing for your microbes. It's also going to be the moisture diaper. It's also going to cut the temperature, and it's going to be most of the bulk volume of your compost pile. There's people that inoculate their wood chips and mature their wood chips. That's great because whatever you put in your ingredients is going to end up in your compost pile to some degree. And then over here on the right, we have beer mash, which is super high nitrogen. Has anyone had old food scraps that have sat out for too long and it smells really bad, right? So what's happening is because of all the available high nitrogen or because of all the available energy in that green or high nitrogen material, you're going to have a huge plume of bacterial growth, especially like, let's say if you're collecting your food scraps in a bucket, if you're a home composter or something, and you've kept that bucket lid sealed, or, in a trash bin situation, you have a plastic liner and the bin's closed. What's going to happen over time is those bacteria are going to use up all the available oxygen in the system. It's going to tilt anaerobic, so non-oxygen breathing, and then all of those anaerobic bacteria blow off the nutrients that you so wanted in your compost pile into volatile organic forms as gas. So that's what you're smelling when you smell old rotting food, is actually bacteria that don't breathe oxygen, that the way that they metabolize their energy is by blowing nutrients off as a form of gas. And you've evolved that sense in your nose to be like, oh, I want to stay away from that because it's full of pathogen organisms. So measuring moisture and achieving correct moisture. If you're going to make a thermophilic compost
Hey,
pile, or
baby,
if you're going to start a
how
compost
was your
pile,
lunch?
I would highly recommend soaking your materials beforehand. A lot of wood chips,
Oh,
especially,
cool, yeah.
can be hydrophobic. Um, and so
Oh,
you really want to give those a chance to,
baseball, soccer.
if I can, there we go. All right. I think I'm still screen sharing. Can you see the presentation on your end? Okay, great. So you want to soak your materials overnight. I always recommend that. And you want 50% moisture. So if you have hay as your green material, how would I get 50% moisture in a bucket of hay? Well, I would fill the bucket up halfway full of water. So half of it's 100% and the other half's 0%. And then when you mix it together, you have about 50. And then you're going to test moisture content using the field capacity squeeze test. I would love to have a demonstration on this live, but unfortunately we can't bring a bunch of soil in here and have dirty hands all over the floor and things like that in this wonderful museum. So I have a chart for you. Ooh. Should work. There we go. Okay, so if you have a handful of soil and you give it a firm handshake, is just about as much as you want to squeeze, and you have a stream of water come out, it's too wet. If you give it a firm squeeze and it falls apart like dust, it's too dry. And you go, okay, well, how do I amend too wet? You add more dry material. And then how do I amend too dry? You add water from your hose. And eventually you'll find about the 50% mark where you want to exist. It's really between 40 and 60. It's not that big of a deal, but it is very important, especially when you first start off. When you squeeze that handful of organic matter, you want about 1 drop of water to come through your fingers. And there's a little bit of tuning with that. Of course, my squeeze strength is a little bit different than someone else's, of course. Oop. All right, so putting the pile together, you have all your ingredients, laid out on a tarp or in your backyard, something like that. You take your pitchfork. If you can notice here, there's water droplets from a hose coming in. Maybe you get your neighbor or your wonderful partner to come help you with this manual labor task, and you guys bond over creating a wonderful compost pile, mixing all those ingredients together thoroughly so they all get to know each other. You're going to put it in your compost receptacle or directly on the ground. So again, really whatever you want to do is okay, especially depending on your feedstock. So the risk here of doing a thermophilic compost pile indirectly is that you're not going to get enough heat in the center of your pile to deactivate pathogen organisms and deactivate seed stock. So if I say I'm going to be a home composter, I'm going to get manure from Sam and hay from Sally, and the hay is full of weed seed and the manure has, sat in the back of the trough for a few weeks, and you don't get up to temperature, suddenly your garden is full of, stomach flu and weed seeds. You don't want that. So depending on your feedstock, you really have to make sure you get to temperature, as opposed to using maybe beer mash and a green material like food scraps. If you can get enough of them, you won't run those risks if you don't quite meet the time and temperature requirements. So it's really feedstock dependent. This thermometer is probably like this big around and 1.5 feet long, and it's stuck all the way to the middle of that pile where the hot center of that temperature is taking off. So here's the hot center of a compost pile. And again, this compost pile is on the ground. This would be the cross section of a windrow, which is how large composting operations operate. They use a big, huge machine to turn the piles instead of moving them by hand. So when you have a compost pile, you want to measure temperature. You want to make sure it reaches this baseline temperature of 131 degrees Fahrenheit. And if it stays between 131 and 150, you can leave it Don't turn it for 72 hours. Let's say you come back in 2 days and suddenly it's at 150 degrees Fahrenheit, but it doesn't reach 160. You can leave it, don't turn it for 2 days. 160, oh my gosh, I need to turn this tomorrow probably. 165, I need to turn this tonight. 170, turn it as soon as possible. So in addition to the risk of, you really creating a rocket and creating a fire and maybe a small explosion in your backyard, this kind of temperature uses oxygen really, really fast. And the whole goal of making your compost is to create a living soil food web that grows oxygen-breathing organisms. So if you let your piles be too hot for too long, they're going to use up all the oxygen in the center of this. And then suddenly you have all those critters that you want to destroy with the perfect non-oxygen breeding ground dead center in your pile. And so when you have a pile like this, if you could think of maybe 3 tires sitting on top of each other, when you turn it, you want to get that hot center out, and then you want to put the top of the pile into the center. So you're doing this, center out, top of the pile, down, bottom of the pile, now into the center, back on top. And so you want to be able to rotate all of the parts of the pile that aren't the center into the center, to make sure that the entire mass of the pile is getting that time and temperature requirements. And if anyone's doing this thermophilic composting, please reach out to me. I can be much more specific and thorough. This presentation does have limits. So here's a video of a pile turn, and I just want you to notice the steam. One of the things that amazes me the first time I made a pile was how hot. 200 gallons of compost is a pretty big, or it's about 1 ton, 2,000 pounds of something that's running between 150 and 170 degrees, really puts off a lot of heat. So you can see the steam coming up. So that is aerobic respiration. Think about your hot breath in the middle of the winter. That's what all those microorganisms are doing in the middle of that compost pile. And so when you turn it, you're doing a bunch of things at once. You're reintroducing water to make sure the water content's correct. You're aerating the center of the pile that's trending towards oxygen lacking. And you're making sure that the rest of the pile has the opportunity to get into the center. And then here's just a picture so no one forgets. Make sure you put a tarp on your pile. So went through a couple emails with a local farmer, and these were kind of my tips for him. So you're going to do some experimentation with your recipe to make sure you hit that first temperature gate, 131 degrees Fahrenheit. Going to pre-soak your materials. It's really, really hard to finish a pile that never had enough water than it is to amend a pile that had too much. You're going to start with thermophilic piles. You need at least 200 gallons of material. Otherwise, you're never going to have enough biomass for the center of that pile to heat up the way that you need it to. The shape's really important. I've failed a lot of piles that were too tall and skinny. You need about a 1-to-1 ratio between pile diameter or footprint diameter and pile height. So in this picture here, right. So this pile diameter is the same as this pile height. It's going to fill out the entire footprint of a standard American pallet. Moisture, between 45 and 55%. How do I test moisture during the middle of my heating cycle? You have to get elbow deep into your pile, and sometimes you need to wear a glove because it will burn your hand. And you're going to pull out a handful from the center, give it a squeeze. Am I too wet? Am I too dry? And then go from there. You can add water to the side and the top just a little bit, but the best way to do it is make sure you have enough in the beginning with soaking the materials, and you add enough water while you're turning the pile. That's the best way to do it. It's about to start being cold, so 200 gallons of material might not cut it. You might need to go to 250 because it's going to be really cold outside here in the next couple months. If you're composting in February, you might, you could do 2 things. You could make the pile ingredients hotter, or you could add mass, or a little bit of both. So it just depends on how much labor you want to put into a 200-gallon pile. Is quite a lot. It really takes 2 people. So you can increase the amount of green, increase the amount of high nitrogen to make sure that that pile stays hot enough long enough when it's, dropping below 30 at night this winter. So the cool down's really easy. After you've turned the pile a few times, hopefully at least 3 times, that gets all of the outside material into the center. You want to wait, you want to keep turning it until the pile no longer reaches 131 degrees Fahrenheit. Otherwise, continue turning the pile. And maturation. So am I guaranteed that my compost feedstock had the full array of soil food web organisms that I need in my compost, that I need in my garden? No. Are there huge differences in behaviors in microbial communities from niche microclimate to gardening zone? Absolutely. So I would highly recommend placing your finished pile at the bottom of the oldest, wisest tree you have on your property, or just where else it's convenient, next to some woods along the tree line. That will allow that you put the pile on the ground with a few sheets of corrugated underneath, and that will allow infiltration of indigenous microorganisms to climb up into that pile and start populating that pile for you. You don't have to do any work. Optimally, you'd maintain the moisture on this maturing pile for at least 6 months before you used it. So, no rules or static composting. I think a lot of you might be familiar with things that look like this. These are perfectly fine. The answer is it just depends. So understanding aerobic versus anaerobic conditions. I can make my thermophilic pile smell just as bad as any of these as I do if I do it wrong, and vice versa. So if you have any of these composting systems in your backyard and they don't smell good, or they're dripping nasty liquid that's attracting flies, add carbon, add brown material, cut back on your high nitrogen material. That's going to fix the problem. And then the compost that and then those feedstocks that are digested by those microorganisms will eventually turn into finished compost. You might not be as fast as a thermophilic pile, but next season, your tumbler will have some great looking organic matter debris in it that's been digested properly. So leachate or liquids, there's something called a worm tower on the market if anyone's tried worms before. So any liquid leaving your vermiculture situation or any worms that you have, or your compost pile for that matter, if anything is dripping, it's too much water, and it means that the system's suffocating. You'll run into gardeners or even marketing materials on some of these aftermarket products that'll say, worm tea, that we put a spout at the bottom of our worm tower because you overwatered it, the substrate suffocated, and then now you have a tap for an infinite amount of anaerobic pathogen organisms that you shouldn't touch other than with rubber gloves to use in your garden. Don't do that. It's got too much water in it. You should never have to use the spout on your worm tower. You should never see any excess liquid coming out of any of the piles that you use. Feedstock, like I said, time is on your side for these composting systems in the backyard, but also you don't want to run the risk of it not having been enough time, and then you as a human come in contact with mammal manures, pathogens. It never got the temperature, and maybe the seeds didn't germinate because they stayed in the right conditions, and suddenly you have pathogen and weed seeds in your garden. You don't want those. Temperatures, maintain a higher proportion of browns, and avoid high nitrogen materials altogether. So just do lawn clippings, just do leaves in your backyard compost. It's going to turn out fantastic, especially if it's in contact with the ground. All those microorganisms are going to be really happy. You're going to get indigenous earthworms in there doing their bioturbation thing, drilling holes and creating soil structure on their own. You don't really need any high nitrogen materials for the backyard composter unless you really want to. Time is your friend and will do the composting for you. So like this situation, this compost tower, if you pull this thing open at the bottom and you can still identify the materials that you put in there to begin with, it's not done yet. Close it up, wait till next season. If you want it to go a little bit faster, we're going to talk about worm composting here in just 1 second. And then, I'm way over time. I'm sorry. Environment, ground contact is key. So when in doubt, worm it out. That's what I always say. All right, vermicomposting. So vermicomposting is composting using worms. These are composting worms. They're a lot different than your earthworms. They're going to eat that organic material. They're going to go after that organic material and they're going to turn it into worm castings for you. Anything, kitchen scraps, pre and post composted materials. If you're insecure about your backyard composting, get some worms. Let them finish it off for you. They're going to be the guarantee for that backyard situation to make sure that everything's going to come out garden safe or production safe. All right, some differences between composting and earthworms, which I went over. We'll skip ahead for respective time. Composting red wigglers. If you want some red wigglers, send me an email. I'll give you some probably. I have plenty, but they're the most common composting worm. You can also buy them online. They're self-replicating and self-population regulating. So if there's too many, you never have to worry about an overpopulation of worms because for some reason they know like, oh, we've maxed out this moving tote that you've put us in. We're not gonna have any more babies. And then the worm surface, worms are kind of slimy. So that residue or mucus on the outside of the worm, as well as on the inside of the worm, is full of beneficial oxygen-breathing microorganisms that are going to directly compete and or destroy any pathogens they come in contact with, both on the outside and on the inside of the worm. So that's why they're such a great finisher for backyard compost that you might not feel too great about. How do you make a worm bin? Millions of hours of content on this online, but the worms need a house. They need a bed, so you can just put some cardboard down in the bottom of a plastic tote. You can put your worms in there, add some food in, about the ratios that you want to start a compost pile in. So maybe 60% of it is leaves, 30% of it is lawn clippings or food scraps or something like that. And you can just dress them. And then you want to put a blanket on top of those worms. If anyone gets worms because of this talk that you heard today, just know that if you order them offline, they're not going to know where they are. And when worms get upset, they go up and out. So if you get worms and you put them into a worm bin, make sure you cover them or leave a light on. Otherwise, when you come back the next day, they'll have exited the bin and then crawled up your walls and dried. It's never happened to me, but I've heard stories. So these are some of the foods that you can feed your worms. Yes and no. So just safe organic materials on the left. These don't necessarily irritate worms like meat and fish, but they're human pathogens. So you don't want to mess with, decaying mammalian meats in your vermicompost, even though worms could handle it eventually. There are some things that do irritate worms. Citrus fruits and peels and pineapples eventually will irritate worms and drive them out, especially if the population isn't really stable. You can get away with all these, but your worms need to be really happy. You need to have a lot of them and you need to use them sparingly, just like, eating some of this as a human. All right, so just some pictures of a worm bin. We have corrugated in the bottom. We have our feedstock ready. We have our browns here to mix in with our feedstocks. You can put them in a moving tote. This person drilled holes in the bottom because they're planning on adding too much water, which you won't do. This is a picture of one of my worm bins just to see kind of like semi-matured. It's got some old substrate blocks in here for mushrooms. It's got that corrugated at the bottom and it's saturated. You want to carry way higher moisture content in a worm bin than you want to do in a compost pile. Think like 60 to 75%. So when you do that squeeze test, you can almost have a stream of water coming out if you really wanted to, just as long as you see some worm cocoons in here. If there's some pregnant worms, it usually means they're happy. Just some different angles of how you might layer a worm bin like this. Super easy to do. Everyone has one of these laying around, bulky item pickup. You can go get a worm bin for free. Fill it yourself. Here's the top of a worm bin with a blanket. That's why I chose this picture. So composting worms like to stay at the top of the soil surface, and they're going to eat everything you put on top, which is really convenient for you because those worms are going to constantly move up, 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 up. Some vermiculture systems you can harvest from the bottom. I harvest my bins left to right. So put all the food in at the right for a few weeks at a time. All the worms are going to move over to the right. You can pull the castings out of the left, then backfill it and start it over. All right, then you can separate the worms from their castings, the casting from the worm. You can buy a nice screener like I have, or you can just make one at home. And this will allow you to separate, the black gold from your vermicompost, turn it into processed worm castings, keep your worms, and then use them again in another bin. So maybe here's a difference between someone that used 10% worm castings versus Scott's Miracle No. And so here's a nice difference between root depth and structure in someone that used worm castings and someone that didn't. Benefits of worm castings, it's what we've been talking about this whole time. Nutrient-rich fertilizer, it's plant available, it's full of beneficial microorganisms, it enhances soil structure, and it boosts plant health and growth. There's also a lot of hormones in worm castings that benefit plants and microorganisms in a lot of different ways that maybe your compost doesn't by itself. Just more reasons to use worm castings. Eco-friendly and sustainable. You can make more worm castings than you know what to do with in your basement and never buy fertilizer again. And you never have to throw out any of your food scraps ever again either. So this is a close-up shot of worm castings. They're these little grainy particulates there. You can do worm composting in your garden bed if you want to. Just take one of those metal mesh buckets, do the layering that I talked to you about, cover it with soil, put some worms in there, come back every once in a while when that layer has gotten digested and it sinks down, fill it with, your kitchen scraps. Those worms will go out into your, maybe not this deep, this is a computer picture, but those worms are going to go out into your garden bed, passively fertilize, and then they're going to get bored or scared, and they're going to smell this bunch of easily accessible food you put in here, and they're going to come back and get this food. Liquid amendments. So you spend a lot of time making your compost, your vermicompost, your worm castings. How does a little bit go a long way? And when you hear of compost extracts or compost teas, they're not made from manures, they're not made from plants, and they're not made from ferments. Otherwise, it would be manure tea, plant tea, or fermented plant juice, FPJ. There's Korean natural farming angle to this ferments one. These are completely different than a compost, an extract tea. So here I'm making compost extract. You can take something as simple as a paint strainer. It has the perfect netting width to let all those microorganisms get through without a bunch of organic material. And you're going to sit there and massage that organic matter into a bucket. So compost extract is different from compost teas in that you're not adding ingredients into the water for the microorganisms to grow off of, and you're not adding air to the system. Here's the compost tea brewer running, and I have a video of that later, but there's a tea bag with compost in it, and then this tube is attached to a pump, and it's actively aerating the compost. There's no reason why every single home gardener shouldn't be doing this every single week in the spring. So easy to do. This is the compost extract. You take a few handfuls of your compost, take a few handfuls of undisturbed forest soil from your backyard, take a few handfuls of your worm castings, and you get a drill with a paint mixer on the bottom, and you just go to town for 30 seconds or a minute in that liquid, and you do a soil drench directly to your plants. Night and day. So easy to do. That drill action is going to get rid of all the, it's going to knock off all those organisms attached to the organic matter, and it will squish some of your nematodes, but it doesn't kill everything. Don't worry, I've verified by microscope. And then I included this bigger one, so I knew there might be some larger scale producers in here. You get a bigger drill and a trash can, you just scaled up your compost extract production a bit. Actively aerated compost tea, instead of putting herbicide in your sprayer, I want you to put actively aerated compost tea in your sprayer. Quality, process, and things like that, we could go over at a later date if you reached out to me. But the key here is, if you ever hear compost tea, If you ever see it at the store, you ever see it at the farmer's market, I want you to make sure that someone ran an air pump through it at least some point. Otherwise, it's not compost tea. It's a leachate. It's an extract. Or it's suffocated. Okay, we got to go. Okay. All righty. Well, that'll be it. Yeah. Thanks. Ooh.
Thank you all for being here. We will be here next week with a wrap up. Again, just check out the offerings for the greenery workshop.
Welcome.
It's a wrap up of like a